to get started with the block review for block 1. And I put the note on the slide for you guys, but this review does not encompass everything that you guys need to focus on. It's a 70% solution, so I recommend, if you see it in here, I recommend that you look over it as many times as you need. That's just my recommendation. You don't have to listen, but it's up to you. All right. So we talked about how to calculate the nutritional values and the calorie content in a food item using the conversion for grams, proteins, and fats. And so when you guys do this, if there's a question like this on the exam, don't be tricked, or don't be fooled. You would calculate, you would take the total grams in that food item and use these conversion factors here in order to determine the calorie content. Does that make sense?
Yes, sir.
And someone just give me the steps on how you would calculate BMI with these equations. If there's a question, the equation will be provided, but what are the steps you need to do? Pounds to kilograms, inches to centimeters. One more thing. Male or female. You guys will know it. I mean, it's obvious,
But you said the equations will be provided?
yes, yeah. So they're different, so that'll be another hand. Well, any questions? Yes.
Will they tell you which one's the male formula? And which
Yeah.
one's the female?
Yeah, he asked, will they tell you if it's a male or female formula? Yes.
Do we know how to convert from pounds to kilograms and inches, or is that?
That will be provided. All right. Yes.
Sir, you do have a hidden slide on here just for the digestion of carbohydrates. We can get rid of that one?
Yeah.
Okay.
So speaking of carbohydrates, here are some examples of monosaccharides. Remember we talked a lot about the different functional groups. What functional group is circled here? All right. What about this one? And this one? It's the achyrocarbon. So remember when I'm kind of getting ahead of myself, but remember when you're trying to figure out if it's an L or D isomer, you take you use the carbon that's the furthest away from the aldehyde group, and it's gonna be the one above the achyrocarbon, the furthest chiral carbon away from the aldehyde group. So if you use this one as an example, 1, 2, 3, 4, 5, it's 5 carbons. This is the achyrocarbon, so this one would be the chiral carbon furthest away. So what isomer is this? D isomer. Perfect. Everybody understand the definition of chiral versus achiral? What's the definition of chiral? It's here, but tell me what's the definition of a chiral carbon? 4 different groups. Hands, always use the hands, that helps. And then if it's chiral, what else is a chiral?
Mirror images cannot be superimposed.
And it's an enantiomer, okay? Does that make sense?
Oh.
What about achiral? Two or more of the same groups?
Sir, just going back to the chiral atoms and such, is it that all enantiomers are chiral, or is it just chiral is an enantiomer? Maybe that's confusing, but like, is an enantiomer a completely different thing than a chiral atom, or is like all enantiomers chiral?
All chiral molecules that cannot be superimposed are an enantiomer, so it's kind of equal. Chiral equals an enantiomer. Does that make sense?
Yes, sir.
Achiral, you said it, 2 of the same atoms or groups around an achiral carbon in this case. talked about the L and D isomer, you guys already know what that is, you just told me. Remember the L isomer, the hydroxyl group is on the left, the D, the hydroxyl group is on the right. Hayward structures. You guys know the difference between the alpha and beta isomer for this one, right?
It's alpha.
Right. Any questions about this?
Fissions.
These disaccharides are very important. You may see some questions about them on your exam. So monosaccharides are linked by glycosidic bonds. We have glucose is one of the monosaccharides that builds the three most common disaccharides. 2 glucose make a maltose, glucose and galactose make lactose, and glucose and fructose make sucrose. And here are the bond types in these 3 disaccharides. Any questions about the bond types? Yes.
Can you go over real quick sort of why sucrose, the glycosidic bond is beta?
Because, so for all glycosidic bonds, well, not for all, but, so you start with the first monosaccharide. Remember, this is the anomeric carbon, that's the carbon that comes from the carbonyl functional group, the C double bond O. So, because for maltose, the anomeric carbon on the glucose molecule is the only anomeric carbon participating in the bond. So that's why you have alpha, because this is an alpha isomer, right? So same thing with lactose. The anomeric carbon is here, and it's a beta. Now sucrose, both anomeric carbons are participating in the glycosidic bond. That's why you have the alpha beta 1, 2.
So that's because it's the first, so it's the first carbon in each monosaccharide.
It's not the first carbon in sucrose, it's the carbon coming from that C double bond O.
Right.
So because both of the carbons participating in the bond, the glycosidic bond, are the anomeric carbon coming from that C double bond O. That's why you have both. The nomenclature is telling you that it's alpha and beta, alpha from the glucose, beta from the fructose, 1, 2, 1 from this carbon, and 2 from this carbon. Does that make sense? Yes.
Sir, and the anomeric carbon in fructose is involved because it's a 5 carbon sugar, not a 6 carbon sugar.
It's just, it's a 6 carbon sugar, but see how 2 of the carbons are outside the range, 1, 2, 3, 4, 5, 6. It's just that structure of the fructose. It's still a 6 carbon sugar, but it's just, it formed a cyclical structure that looks a little bit different. So that's just where the anomeric carbon ended up on fructose. Does that make sense? Any questions? This is just the carbohydrate concept map. Make sure you look over all of the different things that we talked about, including the polysaccharides. So amino acids, we know that amino acids all have an ammonium group, a carboxylate group, and an R group, and the R group dictates the function of that particular amino acid. Now remember, if an amino acid is uncharged, if the functional group is uncharged, then an amino acid with the ammonium group and the carboxylate group will be net charged at physiological pH. Does that make sense? So what would a amino acid that has a carboxylate functional group, what would that be charged at physiological pH? Negative, you say? Correct. Does that make sense to everybody?
Can you repeat that?
So if you have an amino acid that is neutral and these are the only two charges on the amino acid, like this functional group here is neutral. So at physiological pH, these charges will cancel each other out. So it will be deuterium, which is net charge 0. Now if we look at,
Oh, I thought you said negative at the end, that's what.
no, this is what I asked. So I asked if you have this amino acid, what would it be charged?
Oh, negative.
Does that make sense? What about this one? Positive, okay. So remember the different functional groups found in each of the categories of the amino acids. So the nonpolar, they don't like water. They include that aromatic ring, hydrogen, and the alkyl, which is carbon and hydrogens. Polar, they interact with water. They contain amides, thiol group, which is the sulfur and hydrogen, and an alcohol. CHOH is an alcohol.
Sir, and that's on the terminal end. So in order to identify the R group, it has to be on the terminal end of that R group. Or I guess so, like, you would want the alcohol.
Because it's going to be able to interact. That oxygen will interact with another bond.
So if like a nitrogen is like hidden inside, then you would have looked at that to identify what type of amino acid.
No. Yes.
So can you explain the difference between polar and neutral and like non-polar? Like other than just like the, like is there a like should we just remember like the type of functional groups, or is there like a are polar and neutral able to be functional but not polar?
Because this oxygen can interact, like this oxygen is very reactive. And so is that sulfur group. That's why. So for the nonpolar, these don't interact because they're so tightly bound. There's nothing on here that's gonna really interact. Like this one, like I said, this one may trick you, but it's embedded in between an alkyl group, which is not going to interact. The molecule's gonna stay together. That's why it's a non-polar molecule. Any other question?
Can you explain more about the aromatic ring and kind of what that?
The aromatic ring is composed of carbons and hydrogens around the ring structure. That's why it's non-reactive. It's kind of like an alkyl ring, unless you see this one has a nitrogen, but when you see it like this, it's just carbon and hydrogens together.
Technically non-reactive.
Right. Yes.
On the test, just hypothetically, are we most likely to see a picture of the molecule and then be able to decide, or would we just given, hey, it's an alkyl or an aromatic?
Maybe both.
Maybe both.
Okay. Any other question? Any other question? Yes. Oh, thank you, yeah. His question was, will you get a picture and be able to identify, or will you be given a name? I said, maybe both. I don't know for sure. I would know both. Yes. Can you speak up?
Are we expected to know like the name, glycerine, alanine, bailey, like?
Absolutely not. And I'm glad that I don't get to pass that torture on you guys. Because I had to draw all the structures like 3 times in my college career. Whew, I just got flashbacks. Any other questions? No other questions? All right.
We got a question over here.
Yes.
I know you said you don't have to memorize those, but like overall, for the essential amino acids, we do need to memorize them, right?
Just the names for the essential amino acids, I would know the names of them and use that private SIM hall. I would You don't have to know the structure. All right, we talked about the polar acidic. They have that extra carboxylate group here, one here in the backbone and one here, which is gonna make it negative at physiological pH. And then same thing with this polar basic, they have an additional ammonium group, which is gonna make it positive at physiological pH. Talked about the different structure types. What is the primary structure of an amino acid? What does that consist of? Say one person say it loud for my audience.
The sequence of amino acids.
The sequence of amino acids bonded, what type of bond are they? Peptide, what's another name for peptide? Say again?
Amide bonds.
Amide bonds, yeah, that's correct. So we know for collagen, it's a little bit different. It is considered a specialized secondary structure. It's 3 different amino acid chains that are bound like in a rope or braided-like pattern. And this is how connective tissue is formed in our body. And the tight hydrogen bonds is what makes it strong. All right. What type of tertiary interaction is this?
Sorry, I had a question about collagen. So that's like the exception, right? Like it's normally like one strand of peptide bonds and
Yeah.
that folds.
Yeah, this is the exception. And again, if this is on a test, it's not gonna be a trick. It's not to trick you guys. It's just, if it's there, then just know this. No entrapments. I don't believe in that. Say what?
It's like the last guy said that.
Well, you got this. I'm sure you'll tell me, like, I got you guys. I don't believe in getting people guys. I don't want to get guys. So it's just how I live my life. Tertiary structures. What's this an example of?
Hydrophobic.
Hydro what?
Fillet.
Fillet, why?
Interactive.
Hydrophilic because it's interacting with water, right? Does that make sense? What about this? Why is it hydrophobic? 2 hydrophobic groups are forming a hydrophobic core, right? Remember aromatic, hydrophobic, alkyl hydrophobic, right? What about this one? Salt bridge, it's easy, they're opposite charges, so, right? And then this one, hydrogen bond. Alright, see, I want all this stuff. I can just close my laptop and go back to this, right? And then disulfide, how disulfide bonds?
Between
2 what? 2 cysteines, and what's the group? Sulfur, diethyl, sulfur, yep, correct. Any questions about those? Yes.
And we care about disulfide bond, that was for tertiary structures, or was it?
That was for tertiary, that's tertiary. These are all tertiary structure interactions. So it would be 2 cysteine groups within the same chain for the tertiary. Any questions about that? All right, we talked about what denatures a protein. Increasing temperatures to above what? 50 degrees Celsius, yeah. What's an example of an organic compound or heavy metal? Mercury, silver, lead, organic and inorganic compound? Alcohol, yep, isopropyl alcohol as well. All right.
We just need to know, like, should we focus on examples or the bonds that they interact with to the nature of the group?
That table, the table has an example of each and then it has the definition. I would focus on the table. That table, that nice summary table. All Alright, any questions about that? Here are the structures of lipids. What makes all of these lipids? Nature. What else? They are soluble in organic solvents, but not water, right? We talked about all of these except for waxes. What type of structure is this? Right? Saturated. Hey, yeah. Why is it? What does that mean? Saturated.
No double bonds.
Single bonds, right? What are the components?
Is that, I need to know,
Hmm?
is that I need to know because saturated?
I would. What are the components of this fatty acid?
Carbon chain.
Carbon chain, and what else? Carboxylate group, right? Alkyl carbon chain and the carboxylate group. Any questions about that? This is another way that fatty acids can be written, right? Yes, no, maybe? Yes. Okay, thank you.
So the carboxylate group, I thought that had to have a negative charge.
Don't, sometimes it does, sometimes it doesn't, sometimes it has the hydrogen attached to it. Just if you see a C double bond, O bonded to a O, it's whatever else is bonded to it, if it's negative or positive, it's gonna be a carboxylate group. So that's a good question. Mono unsaturated. Why is this unsaturated? Double bond, right? Does somebody have a question? Oh, okay. Well, what structure is this? Triglyceride, right? So we talked about glycerophospholipids as well. We talked about two different types, lecithin and cephalin. They're found in eggs, meat, all that stuff. And lecithin contains choline, cephalin contains ethanol. All right? Yes.
So should we be able to recognize choline versus the two, or just know that there were two types of fatty acids?
I would know that they're the two types. And what are these? Just the last part, what is this? Amino alcohol, right? See that? Nitrogen, the alkyl group, and the OH is bonded to the phosphate group, right? So they're just different length amino alcohols, right? Does that make sense? For ethanolamine and choline? Yes? Please. What type of structure is this? Who? Sphingolipid?
Are you just talking about the bond?
The bond and the structure. What bond is this? An amide bond, correct. And what structure is this? Sphingomyelin, sphingolipid. Sphingolipid is the broad class, sphingomyelin is an example. Same thing for our purposes. Any questions about that? What makes it a sphingolipid? What is this backbone called? Sphingosine, right? Yes? Probably tired of me asking this. What group is this? And this? Amino alcohol. Yep. Alright? Sphingolipid. And sphingomyelin is an example. All right. This is the steroid nucleus backbone, right? what if it had an alkyl group off carbon 17 and groups off 18 and 19? What would it be called? Cholesterol, right? Killing me. Thank you. Right. All right. This is what we talked about with the lipids. Any questions about the lipids? All right, nucleic acids. We know DNA contains cytosine, thymine, adenine, guanine, and a quick way to remember the types of base structures or the base structures is cuts the pie. Pyrimidines are one ring and they contain cytosine, uracil. Thymine, uracil is found in what? RNA. Pure as gold for the purine, the times 2 pure as gold times 2. Purines are the 2 ring structures, as gold, adenine, and guanine. Right? That makes sense. So, guanine and cytosine form 3 hydrogen bonds. What about adenine and thymine? 2. All right. And for DNA replication, what direction does DNA replicate in? 5 prime, 3 prime. Perfect.
Wait, say it again.
DNA replicates in the 5 prime, 3 prime direction. So it's gonna go this direction. So let's just name the components. Actually, that's the name of the components. Somebody tell me the first step of DNA replication. DNA splits how? Helicase. Then what happens?
DNA bonds.
Single stranded. What do? They stop it from going back together. Then what happens? DNA polymerase synthesizes the leading strand, correct, in a continuous manner because remember they're anti-parallel. So this is the leading strand. So the DNA polymerase is going to synthesize that complementary strand in a 5 prime, 3 prime direction in continuous motion. What about the lagging strand? It's a little different. What happens first?
Primer.
Primase binds and does what? Makes RNA primers. And then say it louder.
So the primase needs a little RNA primer that is 5 prime for the DNA, or DNA polymerase that latch onto and goes onto the fragments.
Correct. So did you guys hear your classmate comment? So he said on the lagging strand, primase will make a short RNA primer that signals DNA polymerase to come and remove the RNA primers and make short DNA fragments, which are called Okazaki fragments. And then how the Okazaki fragments join together? Ligase. That makes sense? Any questions about that? And here are the different steps that you guys know oh so well. 3 types of RNA. What's the importance of ribosomal RNA? Say again. Ribosomal RNA combines with proteins, and that's the site of translation, right? Yes?
Yes.
What about messenger RNA?
Bring it to it.
What's the importance of messenger RNA? Template for the peptide strand. Why? What does it carry? Genetic information, and what's the name for genetic information? Codon, right? It carries a codon. What carries the anti-codon? tRNA, right? Does that make sense? Yes. Just knowing what's the intron? What is it? The intron is the junk, right? The exon is what? The codon, right? Intron junk, we don't want it. Exon contains the codon for translation. Speaking of codon, we talked about messenger RNA having the codon, and there are stop signals and start signals. What's the start signal for protein synthesis? What's an example of a stop? Correct. These are bolded. These are bolded. So that's all I'm gonna say. Okay. Any questions? Yes.
For the non-essential amino acids, sir, should we know those?
The non-essential, I would just use or the essential.
The essentials.
Private Tim Hall or whatever mnemonic that helps you. You guys know how to read the codon table? No, you guys have to memorize it. I'm just joking. Absolutely. Come on now, I'm not gonna do that to you. No. It will be provided. So if we're looking for glutamate, the codon for glutamate, it would be G A G or AA, right? So it makes sense how to read it. First letter here, second letter these four, third letter from on this side. So GAA and GAG will give you glutamate. Does that make sense? Yes.
I have a question. So I know you said just remember the names of
The amino acids.
amino acids, right? But then on the table we have the 3-letter and 1-letter, right? So if we're not supposed to remember them, are they going to be provided in the question? Like,
Yes.
hey, you're looking for proline, PRO.
It's gonna be obvious. Again, it's not yeah, it's gonna be obvious. I don't make any talk to me.
So,
Yes.
are we given this exact table? Like it'll say on it, like this one says the start and stop codons.
That won't be on there. Everything else will be on there. Alright? And what is this right here? What binds to here, the receptors? The amino acid, right? The amino acid that matches with this anti-codon loop, right? There are 3 types of point mutations. What's the silent mutation? It's no change in amino acid, yeah, same amino acid. What is a nonsense? It stops, and that's usually very, very severe. I mean, it sometimes results in death. And then, of course, the missense will be a different amino acid that may or may not have the same properties as shown here. These 2 amino acids are different groups of amino acids. All right, questions about that?
Which one did you say leads to death, sir?
This one,
The
typically,
non,
because it's just stopping, like it's going to stop the protein from being synthesized. So, and the damage from missense is variable. Could be severe depending on what the new protein is, or because that's going to affect the folding and all that stuff. Then we have the frame shift mutations. Frame shift mutations either add or delete or insert 1 or more nucleotide. And so what that's going to do is that's going to shift the reading frame. The reading frame is 3 nucleotides together. That's the codon. And so that damage can be variable as well. And so a lot of these are examples of frame shift mutations that I would recommend knowing. Right? We talked about cofactors for enzymes. We know coenzymes are vitamin derivatives. Cofactors are metal ions. Coenzymes are organic molecules. So, someone tell me in your own words, what's happening in competitive inhibition?
There's still a chance
There's still a chance of what?
of that enzyme substrate binding.
Does that make sense to everybody? That was a good answer, that's a perfect.
Thank you, sir.
Still a chance for, there's still a chance for the substrate to beat out the inhibitor, because they're binding at the same site. So if you add more substrate is gonna beat out the inhibitor.
In competitive inhibitors, the Vmax does not change, correct?
We're getting it, yes,
Sorry.
you're getting ahead of yourself, but you're right.
There's no.
Yes, you're right,
Oh, it's right.
you're right. What about non-competitive inhibition? Somebody tell me what it means in your own words. Yes.
So it binds to the allosteric site, it changes the shape of the active site so the substrate can't bind.
And then, perfect, and then what happens?
And then the Vmax.
What happens to the what happens to the reaction?
It gets stunned, so it never reaches the.
It stops, right, correct, the reaction stops, right? Allosteric regulation, we talked about this, negative allosteric regulation occurs when we have enough of a product, we have a negative allosteric modulator that will bind to an enzyme on an allosteric site, change the shape of the active site, so the substrate cannot bind, so the product cannot be made because you have enough of the product. Does that make sense? Alright, so what does Vmax mean? In your own words. The maximum rate of product that can be produced. And why? That's correct, but why? Because all of the enzymes are occupied. Does that make sense?
Can you explain that again?
So Vmax, it means that all of the enzymes have substrate bound to it, so that the reaction, it cannot continue because all the enzymes are occupied. Does that make sense? What about Km? What is Km telling us? Yes, it is 1/2 of Vmax, but what is the Km telling us? One person, I think you're all saying it right, but I'm old.
Well, I guess like how much the enzyme likes the substrate.
How
so
much
higher
the enzyme likes the substrate. So what? So finish that.
The higher the Km, the more the enzyme likes the substrate.
The less.
Yeah.
They're
Right,
inverse.
right.
Yeah.
Inverse.
Okay. So what type of inhibition is the competitive? Why? Vmax doesn't change, but what changes? And so what, the green line, what does this mean? What is the Km telling us in this green line?
It doesn't like the substrate.
It doesn't like the substrate. Make sense? And this is non-competitive inhibition, you guys explained that, so you guys are explaining this so wonderfully. Non-competitive inhibition, the substrate or the inhibitor binds to an allosteric site, it changes the shape of the active site. So enzyme can't bind, but even if it does, the reaction is gonna stop. Hence, you have a reduction in Vmax. Does that make sense? Yes. This one, yes, it's reversible if the inhibitor detaches or if the inhibitor is removed from our system. Yes. That's a good question. Yeah. What's the purpose of the line we revert plot? The overall purpose. It helps us, it's a straight line, so we look at the intercepts and that helps us better approximate actual values. Because this graph, it's a hyperbolic graph. You can't really approximate actual values with this, so, but with this, because the 1 over V max crosses the y-axis, 1 over Km crosses the x-axis, so we can get actual values. Does that make sense?
I got a question.
Yes.
If this graph is better than the other graph, then why do we still use the other graph?
Because they made me learn it. No, I'm just kidding. No, because I think they want to teach like the origin of this, because this is used even now to this day. But you know how they make us learn stuff that is not really pointless. I shouldn't say pointless, but they want us to learn the original means of scientists calculating the Vmax and Km. And you asked a good question the last time, and I was thinking about it. You asked, why do we need the inhibitor? Like, why would we want an inhibitor? And I don't know if this answers the question that you asked, but so when scientists came up with this equation, they were doing all of this in a lab. They didn't have computers, of course, and so they were using these plots. They were coming up with these plots after figuring out after deriving all their data, and this is the plot they came up with. So this is how they determined, they were able to determine that the Vmax remained the same, or if the inhibitor was added. And that's how they were able to determine if it was competitive versus non-competitive, because they were looking for a specific product. Does that make sense? Kind of, maybe? No? Yes?
So just between the, like I was mentioning, and the Limeweaver Burke, you would say the Limeweaver Burke is more accurate to read, it gives a more...
It's easier, it gives you a better estimate of actual values of Vmax and K. So and it's still used in pharmaceutical labs now. We still use this model to determine the Km and Vmax of new enzymes they discover and drugs as well, new drugs that are developed. So, and this is competitive inhibition, and you guys already talked about what happens, but this is the Vmax, right? So Vmax is the same for competitive versus non-competitive. And then the Km is shifted to the right with the inhibitor. So again, that means it can still win, right? It still has a chance. Yes?
So is this saying that Km was increasing more substrate is at the site trying to?
Yeah, so this is no inhibitor, just the substrate. This is with the inhibitor, so it's shifting to the right, so that means more substrate was added and we're still able to reach the Vmax. So this equation, or this representation, red line, no inhibitor, just the substrate, blue line, with the inhibitor, substrate inhibitor. So what this is saying is that when more substrate is added, you still are able to reach Vmax. Does that make sense? What can be said about the Km values in this graph?
Km stays the same?
Km stays the same. Why?
Because you didn't add any more substrate.
One person saying?
It's not competitive.
Your x-intercept is the same, yes. What type of graph is this? What is this?
Non-competitive.
Non-competitive. So what happened to Vmax? It dropped. Why? It binds to the allosteric site, changing the shape. So the reaction, even if the substrate can bind, even at equal substrate concentration, the reaction is not able to continue. Right? Remember the musical chairs analogy? Hopefully this helped you guys. Maybe not, but just say yes, please.
Wait,
Yes.
Thank you.
is the musical chairs gonna be on the exam?
No. Okay. No. This is just like yes.
Label which one has a kid on it.
you might have some questions asking about what does Kenyan mean and all that stuff, and you guys already answered this. You already know it. Right? And what happens in irreversible reactions? Right, right. Enzymes destroyed, it can't come off, your body needs to make the enzymes. Right? Does that make sense? And COX-1 is inhibited by aspirin or acetylsalicylic acid. That's the example. All right, last slide. Vitamins. I recommend knowing this information. This may be on your pants. I'll say it again. Any questions? All right, if you guys have any questions, you can email me. My office is 1319. And have a great week. Yes. The name, the full name, the letter, and remember, there's only one of them that has 4 columns. The rest of them, right? So the name and the function. The name and that function represents a deficiency, except for which one? Nice, right? Okay, I've said way too much. Yes.
Sir, quick question. Going back to the polar acidic R groups on the amino acids, is it called polar acidic because it's given out the hydrogen?
100%. Yeah. That carboxylate donates a hydrogen. That's why sometimes you see it as has a hydrogen. It doesn't have the charge sometimes, but a carboxylate group is considered an acid because it has the ability to donate a hydrogen. So if it's not there, that means that hydrogen is already provided. Very good question. Any other questions? Yes.
Dumb question, sorry, but
No dumb
when you said it basic, they'll be marked with the positive and negative.
Yes. But you already know it. Can you speak up?
You said this review is about 70%. How much does the knowledge check or the check on learning?
I haven't calculated that, but y'all have answered all my questions. So y'all guys are gonna do great. Any other questions? It's hot in this room.
Run while you can.
Do you guys have any other questions? All right, you guys have a good day.